Metabolism is an important area of biochemistry in which biomolecules participate in the reactions of numerous metabolic pathways. Students often have great difficulty in remembering the structures of the main biomolecules of metabolism, mainly because of their complexity and the large number of structures involved. The purpose of this presentation is to describe a new way of representing the structures of biomolecules that makes them easier to remember. Let's start with simple biomolecules, such as occur in ethanol metabolism after alcohol consumption, for example. Using classic Fischer projections, the structure of ethanol can be compared with the ethane gas molecule that occurs in a more reduced state, since it has more hydrogens. Fischer projections provide a way of presenting biomolecules chemically by using symbols to represent atoms and their respective bonds. In this scheme, ethanol is a very simple molecule composed of two carbons, with the alcohol group consisting of a carbon-linked hydroxyl. In metabolism, ethanol oxidation occurs through the enzyme alcohol dehydrogenase to form acetaldehyde, with the reduction of coenzyme NAD+ to NADH. Acetaldehyde has a carbonyl C-bound O referred to as an aldehyde group because it is at the end of the carbon chain. If it were in the middle of the chain, the carbonyl would be known as our ketone group. Acetaldehyde undergoes a new oxidation catalyzed by the enzyme acetaldehyde dehydrogenase to form acetic acid, the major molecule in vinegar, via the reduction of NAD+ coenzyme to NADH. Acetic acid has a carboxyl group that, in solution, usually deprotonates to become negatively charged. In the final step, acetic acid condenses with coenzyme A to form acetyl-CoA, an activated form of acetate that can then enter different metabolic pathways. Frequently, however, life is often not this simple. Take for example one of the most important biomolecules in metabolism, but that is still relatively simple, the glucose molecule, which is a sugar or carbohydrate. In Fischer projections, glucose appears as a sequence of carbons linked to alcohol groups, with a carbonyl group at the end, corresponding to an aldehyde group. Glucose is a hexose or 6-carbon sugar and can be considered an aldose since a carbonyl group is present at the end. We have here the open form of glucose. As can be seen, the carbons are conventionally numbered from 1 to 6, with carbon 1 being the carbon closest to the carbonyl group. Glucose is cyclized in solution, in equilibrium with the open form, which accounts for only 0.25% of this molecule, the remaining 99.75% being in the closed form, but we will not consider this further at this time. For the present discussion, let's consider the open form. A very important glucose isomer in metabolism is galactose. As you can see, the structure is very similar to that of glucose, with the hydroxyl and carbon 4 on the left rather than on the right. Note that this carbon is asymmetrical, or chiral, meaning that all 4 covalent bonds of the carbon are different from each other. Glucose, or galactose, has 4 asymmetric carbons and can form 16 different compounds, depending on the position of the hydroxyls. Returning to glucose, we note that this sugar can be metabolized via the glycolytic pathway, a very important pathway that has 10 reactions. In the first reaction, glucose is phosphorylated to glucose-6-phosphate at the expense of ATP. This product isomerizes to fructose-6-phosphate that is phosphorylated to fructose-1,6-bisphosphate, also at the expense of ATP. After this step, the molecule splits into 3 carbon compounds involving several steps that we will not describe in detail here. Even with these small 3 and 6 carbon molecules, there are many details that are hard to remember. The number of atoms visualized is close to 220. Given the importance of phosphate in glycolysis, the phosphate group can be shown in color, making it easier to see what is happening. Another interesting way to show glycolysis is to use a colored circle containing the letter P to symbolize the phosphate group, PO32-. This notation is often used to enhance visualization of the glycolytic pathway. If this substitution simplifies the visualization of this pathway, then why not extend it to other chemical groups? For this, we can consider hydroxyls, carbonyls, and carboxyls. For example, the hydroxyl group may be represented by a triangle. In this case, we can also consider hydrogen-substituted hydroxyls that occur in phosphate groups. Carbonyls correspond to aldehyde or ketone groups and may be represented by squares. Carboxyls, on the other hand, may be represented by circles. The remaining carbons that are part of the biomolecule skeletons, as well as the respective attached hydrogens, can be omitted without any harm to the final representation of the biomolecules, since we know that the carbons have a total of 4 covalent bonds. We can also place the non-asymmetric carbon bonds in an inclined position, while keeping the other bonds horizontal. The approaches described here allow the construction of a variety of very different representations of the original molecule. Such representations may increase the possibility of remembering the structures of biomolecules that participate in metabolic pathways, in addition to providing more compact and graphic forms of these components.